YouTube channel after. All right, so I am recording. Awesome. Okay, well, welcome everybody to the Human Physiology Mid-Semester Workshop. My name is Stacy DeMertzis, and I am one of the peer tutors for the College of Health and Human Sciences Student Success Center, along with Jason. He's my co-tutor. And today I want to go over with you all ways that you can prepare for the upcoming Lecture 2 exam. We'll go over key concepts that were discussed in lecture. As said before, in the chat, I have posted some links that you guys can look at later. And I have enabled live transcription. So if you guys are able to see the words as I speak, awesome. And without further ado, we can go ahead and get started. So for the upcoming Lecture 2 exam, there's going to be some chapters that you guys have covered in lecture, such as the nervous system, specifically Chapter 7 and Chapter 9, for those of you that like to refer back to the textbook. I put the chapters there as well. The muscular system, which is Chapter 12, and sensory physiology, which is Chapter 10. So my goal for this workshop is to make it as interactive as possible. I am not a professor, I'm not a lecturer. Although I will be guiding you guys throughout this workshop, there's going to be a lot of times where I ask you all questions, and I really want to facilitate a discussion. So please feel free to interrupt me, unmute yourself, share your thoughts, your ideas, and as always, put comments in the chat. You can, whenever you put something in the chat, it's up to you if you want to send it to the whole group, or you can private message me if you're more comfortable with that. Whenever we are on certain slides where I ask you guys questions, we'll go over the answers and we'll talk about them together. Okay, so let's get started. So at this point now in the class, moving from the concepts in the Lecture 1 exam to the concepts now in the Lecture 2 exam, you guys are now moving towards learning about actual organ systems, right? We start off the class learning about like the cellular level, but the nervous system is the first system that we get to learn about. So it's really exciting. But what I always like to do whenever I have students come to my tutoring sessions, or when I was a student and I took the course, whenever I'm learning something really complex, like an organ system, whatever I'm studying within that concept, I always like to think, where is this happening within the body? And I remind myself of what's called the hierarchy of living things. And this is a concept that I think was introduced to you guys at the beginning of the semester. Does this term sound familiar to you all, hierarchy of living things? Perfect. So the hierarchy of living things, the most, see how I want to explain this. I'll start over here on the complex side, organisms, right? We as humans, we are organisms, we are living things. We are made up of several organ systems, right? The nervous system is an example of an organ system. And organ systems are composed of several different organs. Organs are composed of different kinds of tissues, and tissues are made up of different cells working together to perform a common function, right? So the simplest form of the hierarchy of living things are cells. And then cells make up tissues and organ systems and so on and so forth. So that's kind of like the spectrum of the hierarchy of living things. So I have highlighted in purple organ systems because indeed the nervous system is a system. It's made up of these three things that precede it: cells, tissues, and organs. So this image that I have right here, this is typically, if you were to Google the nervous system, or maybe you guys took a biology class in high school, I'm pretty sure this image is very popular if you're just thinking generally like the nervous system, right? And in this image, it's showing the two main divisions of the nervous system. I want you guys to tell me, unmute yourself or put it in the chat, what are those two main divisions?
Is it the peripheral and autonomic nervous system?
The peripheral is one of them. You got that. The autonomic we'll touch on later. But the peripheral, yes, that's one of them. And then there's another one. The two
Is it the
main,
somatic?
not quite close. I'm seeing in the chat, some people are putting the central nervous system and the peripheral nervous system. And yes, those are the answers. So the central nervous system and the peripheral nervous system are the two main divisions. We'll see later how there's actually subdivisions within the main divisions, and it gets like a little more complicated, but we will examine that thoroughly in a moment. But for right now, the two main divisions are the central nervous system and the peripheral nervous system. And I want you guys to tell me, like anatomically, we'll start with the central nervous system. What is the central nervous system composed of?
Brain and spinal cord.
Yes, absolutely. And I'm also seeing that in the chat as well. Yes, the brain and the spinal cord is the central nervous system. Now, the peripheral nervous system, what is that composed of? This one's a little trickier. I see in the chat spinal and cranial nerves. Yes, absolutely. So the statement I have to the right of the image, when it talks about the nervous system having cells in the brain, spinal cord, and periphery, this is kind of like a general statement that's explaining the central versus the peripheral nervous system, right? So if we have cells in the brain and the spinal cord, that's referring to the cells of the central nervous system. And then cells in the periphery kind of means like lateral, right? And if we see here, the brain and the spinal cord are very medial, right? They're in the center. And then the periphery branches out. And we see these spinal nerves branching out all the way outward towards our body. So that's kind of like this general statement. So yes, good work. We got central nervous system, peripheral nervous system as the two main divisions. So let's look at the subdivisions of the nervous system. So you guys were perfectly able to identify central versus peripheral, but now we're going to focus on peripheral because the peripheral nervous system is kind of complex because it has many subdivisions, right? So the first two subdivisions of the peripheral nervous system, we have the somatic nervous system and the autonomic nervous system. So whenever you guys are studying and you see these two terms, think of the peripheral nervous system because that's what they are a part of, right? Let's focus on the autonomic nervous system. The autonomic nervous system is composed of two subdivisions as well. The sympathetic nervous system and the parasympathetic nervous system. So let's say you're studying and you come across the parasympathetic nervous system. You have to think, okay, which subdivision is this of? And hopefully you're thinking of the autonomic nervous system. And then after that, hopefully you remember that it's part of the peripheral nervous system. So it's kind of this sequence. I always recommend, because there's so many names and subdivisions, is drawing out a diagram similar to this. I think there might be ones in your lecture slides as well, or in the textbook. If not, I would recommend creating one on your own, maybe out of your lecture notes, creating like a kind of diagram like this, especially those of you who are visual learners. I'm a visual learner and it's helped me a lot kind of see where things are. Okay, so we were able to identify or kind of have an intro and look at the different divisions of the nervous system. So what I want you guys to do, I'll give you guys about 2 minutes to do this. I want you all to match the function of certain divisions of the nervous system. And so the way I thought this kind of make it easier on you is you can match the number to the letter. So let's say, for example, I'll do the first one. We identified, as a group in this workshop that the central nervous system consists of the brain and spinal cord. So in the chat, you can put like 2A. You match the number to the letter. So the number represents the division of the nervous system, and then the letters are going to represent their function. And so I didn't have room to write like the whole word. So number 4 is the somatic, number 5 is the autonomic, and then 6 and 7 is sympathetic and parasympathetic. So let's match the division with its function. You guys can put in the chat, feel free to unmute, we can talk about it. You guys can work together, collaborate amongst yourselves. So I'll give you guys some time to do that, and then we will go over the answers together. Okay, I see one answer in the chat so far, pairing the central nervous system with letter A, the brain and the spinal cord. And if it makes it easier for you guys just to like type out like all your answers and then just send one kind of large chat message, that works great too. That's why I'm giving you guys about like 3 minutes to collect your answers. I'm seeing some answers in the chat. Great job. Give you guys a few more moments. About 30 more seconds. Okay, let's talk about what we got. So I'm going to see if I can annotate. Okay, so I'm seeing a lot of answers in the chat. Okay, let's see. Actually, what I'm going to do is I won't annotate and we'll actually just go to the next slide. Okay, can you all see the next slide that has the answers? Yes? Okay, perfect. So thank you guys for putting your answers in the chat, and let's go over the divisions of the nervous system and match it to their functions. So we talked about how the central nervous system, and I have my cursor pointing here at the central nervous system, consists of the brain and spinal cord. And we talked about that in our first slide as well, when we were identifying the two main divisions. Second main division, the peripheral nervous system, it's composed of the cranial and spinal nerves. And as far as its function, it's responsible for what we call afferent and efferent signaling. And we will dive well into that in a moment, but I want you to keep that in the back of your head, that the peripheral nervous system functions by sending afferent signals and efferent signals, but we'll dive into that in a moment. So the two divisions of the peripheral nervous system, right, the somatic nervous system, its function is it is responsible for controlling skeletal muscle. While the autonomic nervous system, it's also controlling muscle, but different types. It's controlling smooth muscle, cardiac muscle, and some of our glands within our body. And I want to pause real quick here, and I want us to talk about kind of the difference between the two. So what do you notice about like the name of this division and its function? Like how, let me rephrase this. What's a way to remember that the somatic nervous system controls skeletal muscle, but the autonomic nervous system is controlling smooth muscle and cardiac muscle? What's a way we can kind of remember the difference? I see an answer in the chat. Auto for autonomic kind of sounds like automatically, and then soma starts with an S, skeletal starts with an S. I like that way of remembering it. And I love especially, Tammy, what you said about how auto kind of sounds like automatic. I'm going to ask you all, do we control the movement of our smooth or cardiac muscle? Is that something that we can consciously control? Yes or no? Feel free to unmute, put in the chat. So I see a couple answers in the chat. No, no, no. Yeah, absolutely. So we cannot control, our heart beating. We can't control, smooth muscles often found in our digestive system, and it helps propel our food forward. We can't control that, right? So the nervous system that's sending signals to those muscles to do its job, they're under the autonomic nervous system. And we can remember that because, as Tammy perfectly pointed out, like autonomic kind of sounds like automatic. And indeed, these processes are automatic. We are not deciding when, the muscles in our GI tract move, or we're not deciding when our heart is beating. Perfect. So for somatic nervous system, soma, the root of this word, also means body, right? And so the somatic nervous system is helping our bodily control, right? We can control our skeletal muscle when we choose to walk or run. So that's a way I like to distinguish it. So let's look at the autonomic nervous system for a moment. And we'll examine the two subdivisions, the sympathetic nervous system and the parasympathetic nervous system. And so notice how these two functions are opposite of each other. The sympathetic nervous system increases your heart rate and decreases your digestive activity. And the parasympathetic nervous system is going to decrease your heart rate, but increase your digestive activity. What's a way we can remember the difference between these two? There's a special phrase that you guys might be familiar with. Sympathetic versus parasympathetic. I think it was covered in lecture. Tell me that phrase, or if you have a different way that you remember the difference between sympathetic and parasympathetic. Feel free to share that. You can unmute or put yourself, or unmute yourself or put your comment in the chat. Let's think about ways we can distinguish between the two. We just talked about ways we can distinguish between somatic and autonomic. but how do we distinguish between sympathetic and parasympathetic as far as its function? Are you guys familiar with the term fight or flight?
Yes.
Yes, awesome. And what about rest and digest?
Yes.
Yeah, so fight or flight, what phrase is that associated with? Is it the sympathetic or the parasympathetic? Think about what it means, or think about that phrase and think about the function. What makes the most sense?
Fight or flight would go with sympathetic nervous system.
Yes, absolutely. Thank you, Darian, for sharing. And I also see, yeah, in the chat, a lot of people are commenting sympathetic as well. So fight or flight, let's think about it in terms of function, right? If the sympathetic nervous system is going to increase our heart rate and inhibit our digestion, that kind of makes sense in a fight or flight situation. If we are in a situation that's dangerous and we need to flee or fight back, we want our heart rate increased so that oxygenated blood can quickly pump to our skeletal muscle so our body can move into action. And so when blood is being pumped to your skeletal muscle in this fashion of a high heart rate, it's actually kind of like leaving the GI cavity because it's not like at that moment, digestive activity is like not the priority, right? So the blood is going to disperse to the skeletal muscles so that the skeletal muscles can use that oxygenated blood to move the body in, an emergent fashion and when we're in a fight or flight situation. Parasympathetic nervous system, low heart rate and high digestive activity is kind of the opposite situation. We are in a relaxed state, so we don't need all of this blood rushing to our skeletal muscle. We don't need our heart rate increased. And so that way, we can promote digestive activity during that time. That's kind of a way I like to differentiate the difference. I like to think of those phrases, and that helps me kind of I always think about like the phrase and then the function too, like why would my heart rate increase if I was in a fight or flight situation, or why would my heart rate decrease if I wasn't? So that's the way I like to remember it. Okay, awesome. So as promised, we're going to talk really quickly about the peripheral nervous system. And I mentioned to you all that its function is, it's either sending afferent signals or efferent signals. And these are two vocabulary words that when I took the class, they confused me at first because they sound very similar and they're almost spelled the same. And so it was really important for me to take the time to understand and appreciate the difference between the two. So I want you all to tell me, ignore right now where it says sensory and motor. We will get to that in a minute. Right now, just focus on afferent and efferent. Afferent and efferent signaling, the best way that I can describe it is showing the direction in which a signal, a neurological signal, is traveling. And so signals are either going to travel to the central nervous system, to the brain, or they're going to go out into our body, right? To our somatic nervous system to get to our skeletal muscle, or to our autonomic nervous system to get to our small intestine, right? So I want you guys to tell me in the chat, you can pair the number to the letter. So afferent, where is that going? Is it A, going to the central nervous system, or is it B, going out towards the body? And then same thing with efferent. All right, I already see a comment in the chat from Graciela. Thank you. I'll give you guys a few moments to think, and then we'll talk about it. All right, so I'm seeing some comments in the chat. Thank you all. Looks like a lot of you guys are getting the same answer. That's awesome. So let's talk about it. So number one, afferent, Yes, is going to be paired with letter A. So afferent signaling is in which a signal is sent to the brain, to the to the central nervous system. And then number 2, efferent signaling is associated with letter B. It's going out to the body, whether that's through the somatic or the autonomic nervous system, but it's branching out of the periphery to other cells in our body. And so I have these two phrases here, sensory and motor, and these are kind of like synonyms in a way to afferent and efferent. So afferent signaling is often also called like sensory signaling, or like when we talk about like neurons later, we'll talk about sensory neurons, and then motor is associated with efferent, right? And we'll talk about motor neurons as well. So I want you guys all to tell me in the chat. I have a special way that I like to remember, but I'm curious if you guys have any tips for your classmates of what are ways, because yeah, afferent, efferent, they sound so similar. They're spelled, almost the same. They're both nervous system functions. It can get kind of confusing to differentiate the two. But you guys were able to correctly identify that, the difference between the two. But I'm curious, how do you guys remember that? Like, what are some tips or ways that you can differentiate it? Feel free to unmute or put it in the chat. How do you guys remember that afferent goes to the brain and efferent goes out to the body?
This hint has been used a lot, but afferent with an A, people say arriving, and efferent as exiting.
Yes, I say the same thing. Okay, cool. Yeah, I would say that too when I first took the course and I was like, oh my God, those words are so similar. I would say that too. So A as an afferent, arriving to the brain, and then E as an efferent, exiting the brain. So thank you so much for sharing. So that's a way that little tip that we can use to remember it. Okay, so let's talk about the cells of the nervous system. So I put up again the hierarchy of living things because, again, when we're studying really complex topics in physiology and even anatomy and other science courses, I always like to take a moment and think, okay, at what level is this occurring, right? So I want to talk about the cells of the nervous system. About half of the cells in the nervous system are neurons, and the other half are glial cells. And so neurons are what is known as the functional unit of the nervous system. It communicates signals, and the signal is either electrical or chemical, and we'll go into that in a moment, but it's communicating those signals to other cells, and those other cells might be other neurons, it might be muscle cells if we're dealing with the muscular system, it might be cells in the GI tract, so just cells within the body. Glial cells, we don't refer to them as the functional unit. That doesn't mean they don't have a function. They are just not functioning as far as sending signals to make like things happen within the body. But they have a crucial role in the nervous system, and their job is to surround and support the neurons. And we're going to see later how that goes about. Like, how do glial cells truly help the nervous system function efficiently and effectively? Before we move on, I want you all to tell me, so I mentioned that the neurons were the functional unit. I want you all to define for me, aside from the nervous system, just in general, how do you define a cell? Now, what's the definition of a cell? So a cell, the way to define it is, it's like the most basic functional unit, right? So when we think about neurons in the nervous system are the most basic functional unit, or like the simplest functional unit. So that's something that I like to remind myself too when I'm studying like cells per different organ systems, just to remind myself. Okay, so let's look at the neuron. So a big part of physiology is also reviewing your anatomy. So I have labeled 1, 2, and 3, like three main structures of a neuron. And so what I want you guys to do, feel free to unmute or put it in the chat, which anatomical feature is associated with what? And I have the, after the number, I have the function. So for example, whichever one number 1 is, receives signals. Number 2, it's where the nucleus is. And number 3 is where a neurological signal is traveling. So I want you guys to identify what 1, 2, and 3 are. And then these are your 3 options, letters A, B, and C. Feel free to unmute, put it in the chat. All right, so I'm seeing some answers in the chat. I'll give you guys a few more moments to answer and then we'll review it. Okay, awesome. So let's review. So I'm seeing a lot in the chat that number 1, a lot of you are saying are the dendrites, right? The dendrites receive the signal. And that's absolutely correct. A way I like to remember it is dendrite. So dendra means tree in Greek. And if you look at the image, these branches kind of look like little tree branches. So that's a way that I like to remember it. Number 2, I'm seeing a lot of you put B, the cell body. Absolutely. So this is the body of the neuron, the cell body. And a clue for you to remember it's the cell body is that the nucleus is there. And then number 3, I'm seeing that a lot of you put that it was the axon. And it absolutely is. So this long tube is the axon. And this is where the signal is received by the dendrites. And it's going to travel through that axon. And then right here, this other number 3 that I put here, this term would be axon terminal, right? The end of the axon. And so at the axon terminal, that's where the end of the neuron is going to be. And the end of the neuron is going to form a synapse with either another neuron or another cell. In this image, I have another cell drawn here. I have a muscle cell specifically. And so I want you guys to define for me, what do I mean by synapse? What's a synapse?
A synapse is a gap.
Yeah, so it's a space between a neuron and another cell. And that other cell could be a neuron, could be, in this case, a muscle cell. And so within that synapse, there's going to be a release of what we call neurotransmitters. And neurotransmitters are chemical messengers. And I bring that up because in this image, I have here kind of explaining the pathway of information, right? So a chemical signal is going to be sent here, right? And the dendrites are going to pick that up and receive it. And then as it's traveling through the neuron, through the axon, the signal is actually an electrical signal, not a chemical signal. But then once it reaches the end, and then it reaches that synapse, right? The neurotransmitters, which are these chemical messenger substances they're within the end of the axon. And when the electrical signal travels down, it's going to tell those neurotransmitters, okay, spill out into the synapse and go to the next cell, whether that's another neuron or a different kind of cell. So a way that I kind of like to remember is the signal traveling through the neuron is electrical, but the signal in between neurons or in between cells, that space, that synapse, becomes chemical, because in that synapse, we're releasing the neurotransmitter. Any questions on that concept? No? Okay, perfect. So we'll go on to the next slide. And in this slide, we're going to look at the electrical signaling of the neuron and truly the physiology behind neurons, the electricity, right? So when a neuron functions, right, it wants to send a signal to other neurons, other cells. Many things need to happen. And so right here, I have pictured number one, a resting neuron. And this is what a neuron looks like this is its state at rest, right? We have a bunch of what we call ions, right? They're charged species. And here we have a lot of sodium. That's what that Na plus is. It's a sodium ion. We have a lot of sodium on the outside of the cell. And then on the inside of the cell, in the axon, we have a lot of potassium. That's what that K plus is. It's a potassium ion. If you guys have taken Chem 30A, this might be jogging some of your memory of potassium ions, sodium ions. So when a neuron is resting, there's no action occurring, there's no major signaling. There's a bunch of sodium on the outside, and there's potassium on the inside. And so within a neuron, right, neurons are cells. So indeed, neurons have membranes, right? All cells have a membrane, so neurons have membranes. And so within the membrane, you guys might have covered this for the lecture 1 exam, talking about like proteins within the membrane or certain kind of channels. So in a neuron, we have a few things going on. We have some sodium channels. We've got some potassium channels. We have a sodium potassium pump, and that's responsible for like keeping that natural concentration of sodium versus potassium. And then leak channels, which they're always open. So sometimes sodium's floating in, sometimes it's floating out. Same thing with potassium. But what's key to note here are the gated channels. And these channels remain closed until a stimulus, until a signal is happening, right? So what I want you guys to tell me is that right here, A, B, and C, I have certain what's called membrane potentials. And so I want you to kind of pair, or first, let me ask you, in a resting neuron, what is the membrane potential? Is it -70? Is it -55? Or is it 30? I want you to tell me it in the chat. What's the resting membrane potential? So I'm seeing some comments in the chat. So the resting membrane potential is actually 70 millivolts. That's the membrane potential when the neuron's at rest. But when a signal occurs, the membrane potential is going to increase a little bit. And if it reaches -55 millivolts, we call that the threshold potential. And that means that we can later have what's called an action potential. And an action potential is 30 millivolts, right? It becomes really positive. So what's going on in between these steps? So when a signal travels to a resting neuron, the gated sodium channels are going to open, right? Because it received that signal telling it to open. And a bunch of sodium is going to go into the neuron. And so when a bunch of positive ions go into the neuron, the membrane potential will become less negative, right? It's going to become more positive. And if it reaches this -55 threshold, it will continue on to what's called an action potential, which is like oftentimes it's called an all or nothing experience because it's once it reaches that threshold, it's going to go all the way to about 30 millivolts. So depolarization, listed here number 2, represents that influx of sodium and creating the membrane potential to be more positive, right? Then we reach number 3, the action potential, the peak. We got 30. And then now that the signal has traveled, it's reached the action potential, the neuron is going to want to go back down to its resting natural state. So there's a difference between hyperpolarization and repolarization. I combine them for now just to keep this concise. But basically, it's when it's going to go back down to negative. Hyperpolarization goes a little bit more negative than negative 70 millivolts, but then it's going to repolarize and then get back to that negative 70. And so with with the repolarization, the gated potassium channels are going to open and potassium is going to leave the neuron. And so when positive ions are leaving the neuron, if positivity is leaving, it's going to become more negative, and then we'll then get back down to our resting membrane potential of negative 70 millivolts. So this is how I wanted to explain like the difference. Let me go back one slide. Between like chemical signaling, electrical signaling, and then chemical signaling. So this right here is representing like the electrical activity within the neuron. Okay, so for the nervous system, so those were what I wanted to cover as far as the neuron. Any questions about the neuron before we quickly touch on glial cells?
When the action potential reaches repolarization, does it go back to negative 50 millivolts?
So it'll go to, that's a great question. It'll go back to negative 70. It's going to go back to its resting. So repolarization is basically going from this 30 millivolts back down to 70, right? It's repolarizing to its natural resting membrane potential. This negative 55 will occur again if another stimulus occurs, right? So kind of the cycle is like A, B, C, all the way back down to A. And then let's say another signal arrives, then it'll repeat A, B, C, all the way back to A. Does that answer your question?
So it will pass negative 55 millivolts to get to negative 70.
Oh, yes, sorry. I thought you meant something else. But yeah, it'll go down. There's a great picture in the lecture slides. It kind of looks like a little heartbeat. It's not that. It just, that's the way I remember it. But it's like this. Let me see if I can draw it. Annotate. Tell me if this looks familiar. This. Does that look familiar to any of you?
Yes.
Yeah, so right here we have the depolarization, action potential, repolarization. So yes, it's gonna like re-hit that negative 55 because it's a scale, right? It's like a number line. And then it'll go back to that negative 70 right here. Does that answer your question?
Yes, thank you.
Awesome. Okay, let me go to the next one. So that's kind of like the physiology of the neuron. So before we move on from the nervous system, let's quickly touch on glial cells. So glial cells, we talked about, they support the neuron, right? And they help the nervous system function more efficiently, more effectively. I want to talk about myelin. And myelin are, let me backtrack. So there's two types of glial cells that I want us to focus on right now. There's actually, I believe there's 5 in total, but I want to focus on just 2 right now: oligodendrocytes and Schwann cells. Oligodendrocytes and Schwann cells are 2 types of cells that form 2 types of what we call myelin. And myelin are these fatty sheaths that wrap around an axon. And so what myelin does is it actually helps speed up the propagation of an action potential. And I want you all to tell me how. Like, why is the addition of this myelin sheath, how in the world does that speed up an action potential? I'll give you a hint. This term right here, saltatory conduction, that's the reason why. But what does that mean? What is saltatory conduction? Feel free to unmute or put it in the chat.
Saltatory conduction is when the action potential jumps.
Yes, so when we look at this axon, right, and we have all these myelin sheaths wrapped around, there's little areas of exposed axon. We call those the nodes of Ranvier, the area of exposed axons. And action potentials, they don't like going through myelin sheaths. They only like going through exposed axon. So when you have a bunch of myelin sheaths, let's say there's an action potential that starts off, right? It's going to jump and then continue at this node, and then jump and then continue at that node. If this was a non-myelinated or an unmyelinated neuron, action potentials would be hitting every single part of that axon. And in theory, it would take longer because it wouldn't have this luxury of just leaping to other parts of the axon. So that's how an action potential happens more rapidly. The difference between oligodendrocytes and Schwann cells is that while they're both responsible for making myelin, they make myelin for different parts of the nervous system. So oligodendrocytes make the myelin that we see in the central nervous system, and Schwann cells make the myelin that we see in the peripheral nervous system. A way that I like to remember this is it's kind of alphabetical, like O comes before S, C comes before P, O is paired with C, S is paired with P. That's a way that I like to remember it. If you guys have any other tips or ideas of how you guys remember it, please feel free to share. But that's a way that I used when I took the course. So that is what I wanted to talk about as far as glial cells. Any questions about those before we move on?
Dr. Anand said that the action potentials don't actually jump. It's just what they say for saltatory conduction. So I'm kind of confused.
Yeah, I think the best, I can see why that's confusing. The best way that I like to describe is like, it's not gonna travel through the myelin. It's only gonna occur at the node. So I guess the reason they say jump is, I guess it's because, like, see where my cursor is? Let's say an action potential occurs there. If there wasn't myelin, then it would occur here and here and like slowly down the axon, but it gets to like skip in a way to this area, right? So I can see why that's confusing. But I would refer, if there's, I'm sure there might be a slide in your lecture slides that goes a little bit more in depth than that. I would refer you to those.
Okay, thank you.
Awesome. Okay, so let's wrap up the nervous system before we go on to like sensory and muscles. So I want you guys to answer a few questions so that we can like sum up this concept. So in this image that I have here, I have a neuron and it's connected to a muscle cell. Remember what we talked about with afferent and efferent signaling? Based on this image, what type of signaling do you think is occurring here? Afferent or efferent? You can just put A or E in the chat so we can save some time. Or you can unmute and share vocally. Is this afferent or efferent signaling?
What's the question again? I didn't hear it.
So based on this image that I have here, I have a neuron and I have a muscle cell. So thinking back to the difference between afferent and efferent signaling, like where the neurological signal is traveling to, what does this image represent? Is it afferent or efferent?
Efferent.
Efferent? See Audrey in the chat shared efferent. Yeah, absolutely. And so how do we know that it's efferent? What gave it away?
Because the signal is traveling to the muscle.
Yeah, so it's traveling to our bodies. It's going away from, like, we're not heading towards the brain right now. We're heading towards our muscle cells. It's going away, exiting. Awesome. So number 2, what specific division of the nervous system do we think this neuron belongs to? Think about the divisions, right? We have the central, the peripheral, and then within the peripheral, we have the somatic and autonomic. Which one do we think this neuron would be? Or what part of like which division of the nervous system? This is a motor neuron, I'll tell you that. But think about the division, right? Is this somatic or autonomic? I'll give you another hint. This is a skeletal muscle cell. Somatic. Thank you, Graciela. Yes, somatic, absolutely. So somatic, soma means body, pertaining to skeletal muscle. This is what this neuron would be considered. All right, so since we were able to identify that this belongs to the somatic nervous system, notice how this neuron has myelin on it. Remember we talked about there's two types of cells that are forming myelin, right? If this neuron is from the peripheral nervous system, what type of cell do you think is forming this myelin? Is it an oligodendrocyte or is it a Schwann cell? So we know this is the sympathetic nervous system. We know somatic is part of peripheral. So what type of cell is forming this myelin sheath?
Schwann cells.
Yes, absolutely. So Schwann cells are associated with the peripheral nervous system myelin, while oligodendrocytes are associated with the central nervous system. Awesome. So last but not least, number 4, remember when we were talking about afferent versus efferent? There was 2 other terms that are often used interchangeably with afferent and efferent. What are those 2? Feel free to put it in the chat or unmute. I'll give you a hint. One of them is sensory. And the other one is motor. So which is associated with what?
Is sensory afferent and motor efferent?
Yeah, so sensory afferent, motor efferent. And then earlier we talked about A arriving to the brain, and that's often sensory information, and then E exiting the brain, which is often motor information. Awesome. So those are our review questions for nervous system. So let's talk about the sensory system. We are a little bit short on time, so I won't have us answer this question, but just to give you a kind of an intro and an overview of sensory physiology. Sensory physiology, we're going to focus right now on afferent signaling, sensory signaling. We are not going to be focusing on motor or efferent right now. For sensory physiology, it means that we are delivering afferent signals to the brain, right? And so right here I have listed like the 5 senses, and I think in lecture you guys might have covered like balance or pressure, I think temperature, maybe pain. So I would refer back to the lecture slides as far as which specific senses and sensory experiences that the professor would like you to know. But I have the 5 ones listed here. And so basically for our 5 senses, right, the way we're able to perceive our senses, like let's take hearing, for example, when we're listening to music, the way we're able to recognize and hear the music is through these afferent and sensory signals that are going to the brain. And our brain is going to be able to perceive that information. So let's move on to the next slide. So the way that works, we call it transduction. So we take a stimulus from the environment, right? And what's going to happen is there's going to be certain types of cells within our body called receptor cells. And these cells are in charge of picking up the environmental information. the receptor cells, very similar to the neuron, are going to go through what's called a receptor potential. They're going to become more positive. They're going to eventually, if they reach a threshold, deliver action potentials to neurons, right? We're going from like an environmental stimulus and changing it to an electrical and neurological signal. That's what transduction is. It's going from one energy form to a different energy form. So we're going from environmental to neurological. And so once the receptor cells, I gave the example of hearing, right? The receptor cells within our ears are going to pick up that information. They'll go through a receptor potential. They'll deliver action potentials to the neurons that are connected to those receptor cells. We call those sensory neurons. And it's going to send sensory information to our brain so that we can perceive the sensation that we're experiencing. So what I want you guys to do is I want you to match, same thing that we've been doing so far, is match the number to the letter. And there's a hint. One of them has 2 letters associated with it. So in blue, I have like the concept, and then in kind of like the orange is kind of like an example of what's going on. So feel free to unmute or put in the chat. Give you guys a few seconds to answer this. All right, I'm seeing some answers in the chat that number 1 is associated with letter A, and it absolutely is. So light from an object is something that's occurring within the environment, right? Number 2, a lot of you guys are pairing B and C together. Great job. So a receptor potential, right? I talked about receptor cells, right? A sensory receptor cell, very similar to the neuron, they're going to have ion channels within the membrane, and those are going to open when it's stimulated. Positive ions are going to come in. And remember what we talked about with positive ions entering the cell, right? The receptor potential is going to become more positive, right? Depolarization. And so as it's depolarized, once this transduction of environmental energy to later, it reaches the receptor cells, and then it's going to transform into like a neurological activity. Once it's depolarizing and it reaches that threshold, it's going to form an action potential. And those neurological signals are going to go to the brain, right? Afferent signaling. And we talked about sensory and afferent are often paired together, right? So this sensory information is being delivered to the brain so that we can understand the sensation we're experiencing. So there were a lot of concepts covered in lecture, like taste and smell and hearing, and eyesight is where the visual system is one of them. And so unfortunately, I can't cover all of them. So I wanted to focus on the eyes. One key step when you're learning physiology is review your anatomy. This is an image that that I had when I took Bio 65, and I think it is really helpful to kind of go back to certain things in anatomy or look at the pictures in the textbook or in the slides, just so that we're able to correctly identify the structures. So physiology of the eyes, right? How do we take the stimulus from light that's reflecting off of objects, and how does our brain understand what we're seeing? How can we perceive the sensation? There's a certain pathway. So if we look anatomically, there's a lot of structures in the eye, but right now I want to focus on the cornea, which is where the transmission of light is going to happen. So the light is going to go through the cornea. And then the lens right here, the lens is in charge of focusing images, right? If an image is too far away or too close, the lens is going to work to accommodate that, right? And we'll dive into that in a moment. Once that image is better focused, it's going to hit kind of the back layer of the eye, the retina. And the retina is where we have our photoreceptors. Our photoreceptors are our receptor cells. So I want you guys to tell me there's two types of photoreceptors. What are they? One's responsible for color vision, one's responsible for black and white.
Cones and rods.
Yes, absolutely. And I also see in the chat cones and rods as well. Absolutely. So cones are associated with color, rods associated with black and white. And so these photoreceptors, as we talked about in this slide, right? They're going to go through a receptor potential, they're going to become depolarized, and then they're going to send action potentials through sensory neurons. Specifically here in the eye, we have the optic nerve. And the optic nerve is kind of like the extension of the brain. And so the optic nerve is going to deliver those signals all the way up to the brain, specifically the visual cortex. So that's kind of an example of how you go from environmental stimulus, you hit a certain type of sensory receptor cell, they undergo certain neurological activity, they undergo certain like membrane changes, receptor potentials, and then that's eventually going to be turned into electrical signaling, specifically afferent signaling. See how much. I'm going to skip to this slide right here. So, oh God. So what's listed here under accommodation of lens is also listed here. So we're not missing any information by going to this slide. So I just think it's easier just to focus on this slide right now. So the lens, right? The lens is going to accommodate for images that are too far or too close. And so how does the lens do that for us? The lens is going to change shape. And the way the lens changes shape is based on a few features in the eye, the ciliary muscle and the zonular fibers. And so I don't believe the zonular fibers are pictured in this image. I think I'll add them. And then when I send the PowerPoint, for those of you that answer the survey, which that reminds me, I am going to repost those links. Here's the YouTube channel. Oh, I sent that. Here is the YouTube channel link. And then the link, I don't know why I cannot send the survey link. I will send that at the end once I stop screen sharing. But yes, I will add the zonular fibers. So basically, the ciliary muscle pictured in blue, ciliary muscle, and then the lens. So when the ciliary muscle contracts, the zonular fibers, they're going to do the opposite. And then the shape of the lens is going to be dictated based on what the zonular fibers do. So I have them highlighted here in the same color so that you guys can follow along. If the ciliary muscle contracts, the zonular fibers are going to relax. And if the zonular fibers relax, the lens is going to become more round. If the ciliary muscle relaxes, the zonular fibers will do the opposite and contract, and that will then flatten the lens. So that's the pattern. And I have them highlighted in the same color. So like ciliary muscle relaxes, zonular fibers contract, lens is flat. So that makes it a little easier to see. So kind of to think about this, when an image is far away and we need to accommodate for that, the lens is going to be flat. We want our lens a little bit more flat when we need to see images far away. And so when we know that, we can think, okay, if the lens is flat, what does that mean for the ciliary muscle and the zonular fibers? What did they have to do for the lens to become flat? Can somebody tell me?
Did they have to relax?
So for a lens to become flat, the zonular fibers have to contract. And that means the ciliary muscle is relaxed because the zonular fibers and the ciliary muscle do the opposite. I lost my chat for a moment. Okay. Perfect. Oh, let me go back. Oh, why is it not? Okay. Yeah, awesome. So I would say the key takeaway for this is that whatever the ciliary muscle does, the zonular fibers do the opposite. Really quickly, I'm keeping an eye on the time. I think I might have. Okay, I don't think we will have time to go over right up here the clinical application, but there is a great slide in the lecture slides that talks about hyperopia, which is farsightedness, and myopia, which is nearsightedness. Basically, that means that a person who's farsighted has a smaller eyeball than someone who is nearsighted, and so therefore the light is not going to hit directly on the back of the retina as we want it to. And because the light isn't focusing directly on where it should, that's why some images may be blurry. And so there's certain lenses that can correct that. But I will refer you to the lecture slides on that because I do want to get through the muscular system before we log off. Any quick questions about sensory physiology before we move on to muscular? And actually, similar to the review questions we had for nervous system, there's going to be a slide where we answer some questions about sensory and muscular physiology. Any quick questions before we move on? No? Okay, perfect. Okay, so let's talk really quickly about the muscular system. So opposite to sensory physiology, the muscular system is responsible, it's experiencing the efferent or the motor signals from the nervous system. We're not traveling up to the brain. The brain essentially is traveling to us, to the muscles, right? It's sending that efferent, that exiting signal from the brain to the nervous system. And so, to the muscular system. And so within the muscular system, we have three types of muscles. We have skeletal muscle, cardiac muscle, and smooth muscle. Today, we're going to focus on skeletal muscle only, but I wanted to point out that there's also cardiac and smooth muscle. And we were able to identify earlier in the workshop that skeletal muscle is under voluntary movement, right? It's controlled by the somatic nervous system, while cardiac and smooth are under involuntary movement. They're controlled by the autonomic nervous system. Couple of vocabulary words when it comes to skeletal muscle physiology are muscle fibers and neuromuscular junction. We'll look at neuromuscular junction in a second, but I want someone to tell me, what is another name for muscle fiber? What's the synonym for muscle fiber?
Muscle cell.
Yeah, absolutely. So that's just another name for it that is important to note, because sometimes I've noticed in a lot of biology classes I've taken, some say muscle cell, some say muscle fiber. So I always advise students to remember that they're the same thing. Okay, so the physiology of the muscular system. So I hope this image looks familiar to all of you. So this is a motor neuron that is innervating with a muscle cell. And so this area right here, right? I took these 3 bullet points from the first time we saw this image, right? The axon terminal, the end of the axon, it's forming a synapse with a muscle cell. We'll see physiologically a chemical signal being sent, a chemical messenger. But let's look at the specifics now. Now that we're not talking generally about the nervous system, let's talk specifically about the muscular system. So the area between, or just this junction of an axon and a muscle, what do we call that?
Neuromuscular junction.
Yeah, absolutely. So we've got the neuron and the muscle. It's also called a motor unit. So when you have a nerve or a neuron that's innervating a muscle cell, it's also called a motor unit. And so physiologically, we're going to have action potentials within the neuron, and then eventually action potentials within the muscle cell. And in between that, right, we're going to have that, going from electrical to chemical, back to electrical. Action potentials refer to like electrical signaling, right? In between, we're going to have chemical signaling in the synapse, right? And we talked earlier about how chemical signaling is neurotransmitters. Acetylcholine, which is abbreviated ACH, is the neurotransmitter often associated with the muscular system and muscle physiology. And so the point of the muscular system is the nervous system is going to send a signal to the muscles so that they can contract, so that our bodies can move, right? Specifically for skeletal muscle. So this will be the last activity that I'll ask you all to do because we do have 10 more minutes. I am willing to go over a few minutes, but I know I want to respect all of your guys' time. So if you have to leave at 6, feel free, but I'll stay on a few extra minutes so that we can answer some questions if you guys have any. So as far as physiology of the muscular system, really quick, let's review the anatomy. So we talked about how a muscle fiber is also called a muscle cell. Within the muscle cells, we're going to have myofibrils. And then within myofibrils, we have myosin and actin filaments, right? There's 2 types of myofilaments, myosin and actin. And when we if we were to look at an image of the myofilaments together, they're kind of sectioned off, right? And we call those sections sarcomeres. And these sarcomeres are the contractile unit. So what do I mean by that? What I mean is that when we think of muscle contraction, right? Automatically, let's think about muscle cells, because remember, the definition of the cell is the basic functional unit, right? And the function of the muscles right now is to contract. But within the muscle cell, we have to keep in mind, it gets a little bit more specific, right? We're specifically looking at the myosin and the actin, the myofilaments. And so that's going to be our contractile unit. So I have an image here that kind of talks about right here in green, we have the myosin. In these blue dots, we have the actin, so the thick filament of the myosin, the thin filament of the actin. And this is kind of representing what goes on in muscle contraction. And so what I want you all to do is I want you to put in order. Right here, I have a list that's out of order. And the next slide, it shows the correction. I want you to put in order what happens. Like, what's the physiology of muscle contraction? Feel free. You can list, like, if you think A is number 1, you can put 1A and kind of live on. There's 5. So I want you guys to put in the chat what order you think this is, and then we'll go over it. Okay, I see an answer in the chat. I'll give you guys a few more seconds, and then we'll quickly go over the answer. And then I need to resend the link for the survey. So I'm going to have to stop sharing my screen while I do that. So we will quickly go over this answer, and then I'll go ahead and stop sharing my screen, and I'll give you guys the link. And then we'll debrief. Yes, a few more moments. Okay, let's quickly go over the answer. And for those of you that complete the survey and receive the PDF of this, I will add extra comments on here, and I'll put them in red colored text so that it stands out. Just kind of an explanation, because unfortunately we don't have enough time to like really deep dive into this. But basically, the correct order physiology of muscle contraction is that we're going to have this depolarization, right? The nervous system is going to get excited. It's going to, undergo action potentials. And so the muscle cell, as it's being depolarized, what's specific to the muscle cell is that it's going to release calcium. And calcium is going to bind to this protein right here. It's drawn in yellow called troponin. And troponin is a protein that binds to tropomyosin, which is something that blocks actin from interacting with myosin. And that's a problem because actin and myosin need to interact, they need to touch, and they need to slide past each other in order for contraction to occur. So if tropomyosin is in the way, contraction can't occur. And so the way that we can then go around that and make contraction occur is it starts off in the nervous system. We're sending a signal to our muscle cells. The muscle cells will release calcium. Calcium is the key. Calcium is the key to get to troponin. Once calcium binds to troponin, tropomyosin's like, all right, I'll move. I'll move out of the way. And then actin and myosin can then form a cross bridge, right? And the way they form a cross bridge is when the actin and the myosin heads come together, and ATP, which is also known as energy, helps make that happen. And so now they're in the position, they're ready to slide past each other. And when they slide past each other, the sarcomere shortens. And that's what muscle contraction is, right? Contraction, we shorten our muscle. So the ATP that's used to get the myosin head ready, and then it forms with the actin, it's going to drop off a phosphate group because ATP is adenosine triphosphate, which means it has 3 phosphate groups. Once it drops off a phosphate group, as we see here, it goes from it's going to release that phosphate group. That release of energy is like power, right? We call that the power stroke. And then the filaments, the myofilaments, can slide past each other. And that's what that contraction is. Any questions on that? Any questions on muscle physiology?
So the sliding of the myofilament is the sarcomere getting smaller, shortening.
Yeah, absolutely. Because the sarcomere, remember we mentioned, are like the sections of the myofilaments. That's the contractile unit. So if the myosin and the actin are sliding past each other, if they're like this and they slide past each other, they're going to get shorter. I mean, the sarcomere itself is going to get shorter. And I can add another picture to this slide as well that kind of better shows that. And I believe there are images in the lecture slides that show it as well. Does that answer your question?
Yes, it does. That helps a lot. Thank you.
Awesome. So these are the wrap-up questions that unfortunately we will not have. Actually, we might. Let me just really quickly stop sharing my screen. So that I can repost the link in the chat. I'm so sorry that I was having trouble doing it from sharing my screen. But here's the link to the survey. If all of you can fill that out, that would be amazing, so that we can get some feedback. And then we got about 3 minutes. Let me go back to sharing my screen real quick. Okay, can you all see the full screen again?
Yes.
So basically these review questions, they're very, very, very similar to the review questions we did on the nervous system. So it's basically asking like the difference between afferent and efferent, asking ways like we remember afferent versus efferent, and like which specific division of the nervous system is responsible for what. So gonna think of a creative way to kind of combine all these questions. So basically, I want you to tell me sensory physiology versus muscular physiology. Which one is afferent and which one is efferent? You just put in the chat or unmute yourself. Sensory versus muscle, afferent versus efferent. Which one is sensory? Which one is muscular? Yep, sensory is afferent, muscular is efferent. And so a way to remember that is sensory, right? We're sensing things, our brain is perceiving it. So the signal's got to go up to the brain. But muscle, right? It's our specifically skeletal muscle, our body's moving. And so that is going to be like a motor effect, right? So the signal, our brain has got to tell our muscles to act, to function. Awesome. And then I'll put the answers to each of these 4 questions in red so that you guys have them. So thank you guys so much. I'm so sorry we're a little bit over time, but good luck on the lecture exam 2. As I talked about in the beginning of the workshop, I always recommend writing things out as you read them, maybe drawing some structures, especially if like there's an anatomy concept that you have to review. And then repetition over everything, study in advance, and be able to review concepts as many times as you can. That's my advice. Just a reminder of our tutoring services. I am not the only Bio 66 tutor. My co-tutor is Jason, and here is our flyer with both of our hours and instructions on how you can make an appointment with us through Spartan Connect. And I believe these flyers are also in your Canvas page. I think Dr. Anand has them posted in the tutors and clubs module. So these posters are there for you guys to look at as well. I have an extra bonus slide. We could not cover the ears this session, but I have some anatomy drawings for you all in case you want to look at those. And then if anyone wants to stay back and ask questions, I'll be here for a few extra minutes, and then we can use this slide to kind of type things out if we need to. So I'll leave my flyer here. Thank you all so much. I hope everyone was able to get the link to the survey. Does that link work for all of you? Yes? Okay. Thank you guys so much. I hope you guys all have a great day. Feel free to stay back and ask questions, and good luck on lecture 2 exam. I know you guys will do great. Study your notes, your slides, watch any of the videos. Oh, also, I forgot to mention, I will be sending also a kind of like a practice exam study guide that my co-tutor Jason created. So for those of you who fill out the survey, you will also be not only receiving the PDF of these slides, but you'll be receiving that practice test with an answer key so you guys can have some more practice and quiz yourself.
You said that the sarcomeres are made up of sections of myofilaments, correct?
Yeah, let me draw that out. Let me do that because I think that would make the most sense. So basically what I mean by that, and I think there's pictures in lecture as well. So when we have myofilaments, right? Like let's say this is a myosin, and that's gonna be the actin because I need to make myosin thicker, right? So here's myosin, there's actin, here's myosin again. Oops. Actin again. The sarcomere is basically, and if you're familiar with Z lines, I think that was a concept in lecture. If you see these red lines here, these are like the Z lines. Each little section, right? I'll put S for sarcomere. So this is what I mean by like the sarcomere is like the sections of the myofilament. So this is like one long muscle fiber, but within the muscle fiber, we have the myofilaments. But then if we see these like little segmented lines, right? And that's how skeletal muscle ends up being striated. That's what we mean by sarcomere. Does that answer your question?
So each of those horizontal stripes, is that a myofilament? I'm confused. Oh no, wait, I think I get it now. Is like the whole line across horizontally, that is a myofilament, and the sarcomeres are breaking up
Yeah.
myofilaments, creating sections, right?
Yes. So these red lines right here are represent like that's I try to draw like a, what are those called? Brackets. That's what this like red squiggly one is. Let me change the color to make this clear. I'm gonna make the Z lines or the sections of the sections of the sarcomere pink so you can see it better. So like this would be one sarcomere, this would be another sarcomere, right? The sections of this long myofibril. There's a slide on, I don't know what number it is, but if you go onto the lecture slides and there's one, whenever it talks about sarcomeres and Z lines, I think that might help answer your question as well. I'm looking in my notes for when I took the class, and it says right here, sarcomere structure. It's a section of a myofibril. It's the pattern of the thick and thin elements, and they're anchored by the Z lines. So if you can find a slide in the lecture, maybe in the textbook with those specific words, I'll put it in the chat for you. Okay, why can't I type in the chat? Oh, because I'm on annotation. Let me, Z lines, sarcomere, what else? Yeah, those two. Those are like your two keywords that if there is a slide that talks about that, I think that might help clear up any confusion.
When will the YouTube video be posted?
Well, I'm not too sure when. It should be sometime soon because I'm going to stop recording and then send in the video, and then the college will then post it to the YouTube channel. But for those of you who answer the survey as soon as possible, I'm going to start right away sending out the PowerPoints to those who complete the survey, as well as the practice exam that Jason created, or the study Jason created.
Thank you.
Thank you so much. Have a great day.